Excellent. All right, folks, so we'll make a start. So thanks for everyone coming out today. It's nice to see a few different faces in the audience. I guess this happens when we mix up the contents of the seminar series. So I guess one thing I've been thinking about whether I should do hosting these sessions or not is whether I should be doing the acknowledgement of country or hand it over to the presenters to do it, but I've decided that I'm going to start doing it. So just like to start with an acknowledgement of country. So acknowledge that we're on the lands of the Larrakia people and pay our respects to their leaders, their elders past, present and emerging. And the other thing that I think is quite interesting or useful to do in a series like this one, where the presentations are often about environmental science or environmental management, is to acknowledge that the Larrakia people were the first environmental scientists and environmental managers on this land. So I had a chat to Anna earlier today. Actually, I probably should introduce people before I start riffing. So today we've got two of our very own from Real. So we've got Dr. Anna Padovan and Professor Karen Gibb. And so earlier talking to Anna today about the introductions, and Anna told me not to worry too much about it. So I've often been told that the shorter the introduction, the higher quality, higher caliber are the presenters. No. But I mean, just as a really short background, so Anna and Karen are in the environmental microbiology and chemistry unit. They both have extensive experience in microbiology, so environmental microbiology. And as we'll hear about today, this is in a marine setting. So I know that they've done work, come on in folks, come on in, to do with recent work on the Darwin report cards. And I think today we're going to be hearing a lot about so there's chairs up the back, anywhere you sit's fine, work relating to oysters and vibrios. So that's more than enough from me. I'm going to hand over to Anna to start. Over to you.
Rightio. Thanks everyone for coming today, and everyone I think over there in Zoom land as well. I'm going to start the talk today, giving you a brief introduction about vibrios, who they are, what they do and why we're interested in them, and then summarise some of the work that we've done over the last decade on these organisms, identify a few gaps that are still remaining, and then hand over to Karen to finish off with her insights and also to provide a vision that we have on this vibrio research into the future. Not that arrow.
I need to click on the screen first.
Rightio. So vibrios are a genus within the bacterial group Gamma proteobacteria. They're small rods, they've got flagella when they're in a liquid environment. Most of them require sodium to live. Some of them have low sodium requirements and can actually live in fresh water. They're primarily aquatic organisms and are very common along coastal areas and estuaries. They can be associated with marine animals, and the photo on the bottom right shows a vibrio called Vibrio fisheri that has a symbiotic relationship with a squid, and it attaches to its light organ to help it see at night. The vibrios can have complex life cycles. Normally, this is in relation to adverse conditions. They can be free living, they can be epibiotic, attached to plankton. I already mentioned they can have an association with animals as well. They can form biofilms, and under adverse conditions, they can form these viable but not culturable cell states. And this is where the cells shrink and they turn into little spheres, and they're called microvibrios. And that's normally when there's a lack of nutrients or under cold conditions. They have a pretty amazing metabolic capacity. They can produce a variety of extracellular enzymes. They can degrade DNA, RNA, proteins, chitin, a whole bunch of things that I've written on there. And they also produce biologically active molecules, such as hemolysins and toxins. Out of the 100 or so vibrio species that exist, there are about a dozen that are pathogenic to humans. And these species or strains within species carry virulence genes and factors that make them pathogenic. And there's also a set that are pathogenic to marine animals as well. So the table here is taken from a Baker publication in 2018, and it lists the dozen or so Vibrio species that are pathogenic to humans. The way they enter our bodies is either orally through seafood that is raw or has been lightly cooked, that is contaminating the seafood, or through wounds when entering marine seawater. So looking at the table there, Vibrio cholera is on the top, and that's the organism that causes cholera. That's not the one that we're interested in today. But there are other cholera strains which can cause gastro and wound infections. And the other big two Vibrio species are Vibrio parahemolyticus and Vibrio vulnificus. So these three together are responsible for most of the seafood-related gastro cases anywhere. The wound infections and also from very severe gastro cases can also lead to septicemia. So they can have serious repercussions. Not every single organism of one species is necessarily pathogenic. So for example, for Vibrio parahemolyticus, most virulent strains contain a couple of key hemolysin or genes, TDH, which is a thermostable direct hemolysin, and a TRH, which is related to it. And these genes and products are present in most clinical strains. However, they're not present in all clinical strains. And in fact, most environmental strains lack these particular genes. So there's suggestions in the literature that there are other virulent genes more so than these two main ones. And they are related to secretory systems. And what these systems are is proteins that effectively inject, they're like a syringe that inject toxins into cells. So for environmental samples, sometimes virulence genes such as VOPC and VOPP, they're just names that are given, are used to detect virulent strains rather than the clinical ones. So around the world, Vibrio illnesses are increasing, and it's been suggested that it's due to global warming. So the graph on the left shows the number of Vibrio infections in the USA over the last 20 or so years, and you can see the trend is upwards. And the graph on the right, taken from the same paper, is air temperature, and we've all seen various permutations of this over the last few years, showing a trend to increasing air temperatures. And so there's a lot of publications coming out over the last 5 or 10 years saying that Vibrios are the barometers of climate change, and their levels are increasing due to sea warming, and therefore infections are increasing as well. In fact, also in Northern Europe, the trend is the same, with the cases of Vibrios increasing, particularly so during heat waves when more people are entering the water and trying to cool off. Overall, Vibrios don't form a big proportion of the total number of food-related illnesses. However, they do account for a lot of economic loss because, for example, a lot of hospitalisation is required for some of the Vibrio diseases. In Australia, there are also cases of vibriosis, either from gastro or from wound infections. And this is a paper that came out recently, showing over the last 15, nearly 20 or so years, the cases from foodborne Vibrio gastro cases in Australia. And similar to the graph I showed before, you can see that there's an increasing trend over the last 5 years. There was a spike here in 2016, and that was due to Vibrio parahemolyticus in Tasmanian oysters. They had an unusually high sea temperatures over the summer, which doesn't seem like a lot. It's about 18 degrees, but that was a couple of degrees above average for their temperature in the water. And there was a spike in Vibrio parahemolyticus in the oysters that led to an increase in cases. And not shown on that graph, because it only happened as recently at the end of last year, there were a number of gastro cases caused by Vibrio parahemolyticus that were tracked back to Coffin Bay oysters. And so they were recalled, and that affected people in quite a few states because the oysters had already been distributed. The reason they knew it was Vibrio parahemolyticus is from detecting it in stool samples. but they weren't actually able to measure it in the oysters. So it's still a bit unclear as to how the vibrios came to accumulate in the oysters. Was it environmental or was it some sort of post-harvest temperature abuse? And in the NT, we also have cases of vibriosis. Here, it's necessary to report both wound infections and also gastro cases from whatever vibrio species happens to be causing it. And this table is from the NT CDC with vibrio cases from food poisoning and also the invasive or wound form over the last 8, 9, 10 years. And you can see the numbers aren't very high, but nevertheless, almost every year there's a case from either gastro or wound infection. And the next, when I press this button, a photo is going to slowly come up and it's a picture of a wound. And so if you don't like seeing that sort of stuff, you might look out at the pandanus. But this is a wound infection. It came from a paper by Anna Ralph and Bart Curry. And this was a paper that they produced in 2007. And it described a case of 3 fishermen in the north and east coast of the NT, that over a course of a few years, there were 3 separate cases of vibriosis, and they were wound infections. And this particular case was a man that had been in water fishing for prawns and I don't know, barra, I suspect, and a wound became infected, and he very...
Ask a question. Is there a plastic cover on that, or is that just the wound?
I think that's the wound. It's got a fancy name. I don't know if anyone else, but it's just a blistering that's caused from the infection. So yeah, so it's pretty awful. Anyway, it has to be cleared off, so that tissue is gone. It doesn't come back again. And in worst cases, it can cause septicemia, gangrene, and lead to amputation. And in patients that are immunocompromised or have some sort of underlying health issues, can lead to death. So I didn't want to be dramatic and scare everyone. Still go fishing, but this is what can happen from. This is a Vibrio vulnificus infection. So we became interested in vibrios back in 2015 when we were asked by Param Water Corporation to do a research consultancy looking at aquatic foods in Darwin Harbour. And they were interested in knowing what the safety issues were around aquatic foods that were harvested in the harbour, especially in relation to sites where treated effluent was discharged. And just in a nutshell, we tested 4 different species: longbuns, mud crabs, oysters, and also mangrove snails, and tested them for E. coli, norovirus, salmonella, and vibrios. But what was most interesting to us was the fact that we found that both Vibrio parahemolyticus and vulnificus were quite prevalent in the shellfish, especially in the wet season. This was a presence-absence, not an enumeration. So for Vibrio parahemolyticus, it was found in 80% of samples in the wet and in 48% of samples in the dry season. It was a similar story for Vibrio vulnificus as well, more prevalent in the wet compared to the dry, but in less samples. We also detected virulent strains. So we detected these TRH or the TDH genes in 12 of the samples, and that represented about 3% of the population. No, 3% of the samples that we tested. And because of these results, we were interested to know, were there vibrios? Well, of course there were vibrios in the water, because I said that at the beginning, but how abundant were they? And did they vary with the seasons? Vibrios like warm conditions. Here it's warm all year round. So do we find them here all year at the same level? So we did a study in 2018 in Darwin Harbour where we collected water from 3 sites, from Ludmilla Creek, Rapid Creek and Buffalo Creek. And we also measured a range of phys-chem parameters. And we filtered the seawater and captured the bacteria on the membrane and then extracted the DNA. So we were using molecular tools to determine the amounts, the levels of Vibrio parahemolyticus, vulnificus and cholera, the big 3. And we also wanted to look beyond these 3 species because there are other Vibrio species that are pathogenic to humans and animals. So we used an approach that had just been developed by colleagues at the University of Technology, Sydney. They didn't develop the amplicon sequencing technique, but they developed the way of targeting a particular gene, which was very good at resolving Vibrio taxa, which other genes that are generally used, like the 16S gene, aren't very good at resolving. So some of those results are shown here. So firstly, the phys-chem, the nutrients TN, total nitrogen and total phosphorus were highest in Buffalo Creek, which wasn't a surprise given that it receives treated effluent upstream from Leanya Sanderson. Conductivity was lower in the wet season, again, no surprise given the rainfall in the wet season. Temperature varied from 24 to 32 degrees, so a narrower range than has been reported in more temperate climates. We also measured DO and chlorophyll A, and I can't remember if there was anything interesting with DO. And chlorophyll A was higher again in Buffalo Creek due to the nutrients and the often algal blooms that occur there. So these graphs show quantitative PCR data where we target a gene that is specific for that, that species that we're looking at. So this showed that Vibrio vulnificus, which is the top graph, was detected in many of the samples, and levels were higher in the wet season, which are the blue samples, compared to the dry, except for a one-off August sampling in Buffalo Creek, where there was something odd happening in the creek system. Vibrio parahemolyticus was detected in all the water samples that we looked at, and again, levels were higher in the wet season. And Vibrio cholera was only detected in Buffalo Creek, not the other two creeks, mainly in the wet season, but again, some detection in the August period. We also screened for virulence genes for Vibrio parahemolyticus and cholera, and no virulence genes were detected in these water samples. So this slide shows the results from the amplicon sequencing data, which enables us to look at the whole Vibrio community. And using this approach, we were able to identify 42 different Vibrio species in the seawater samples, which is about double than has been reported for temperate areas, including Sydney Harbour. We were able to identify also the big three in the data set, which was good. So cholera in the red, Vibrio parahemolyticus in the yellow, and vulnificus in the black. And it really reflected the similar results to the quantitative PCR results, that they were more abundant in the wet season compared to the dry season. To try and understand what environmental factors drive the abundance of the Vibrio communities, Miriam did some multivariate analysis on our behalf, and the plot on the left is a way of showing this complex data in a way that makes a bit more sense. And so what it shows is each of these markers represents a Vibrio community from a particular water sample. So communities that are close together are more closely related, and markers that are further apart are more dissimilar. The red markers are from the dry season samples and the blue from the wet. So you can already see a nice discrimination between the wet and the dry season samples. And overlaid onto these vectors with the variables that best explain the variance in the Vibrio communities. So for Vibrio parahemolyticus, it was more abundant in the wet season, and temperature was the biggest driver explaining its variance. In contrast, Vibrio vulnificus, which was also abundant in the wet season, its variance was best explained by total nitrogen and also total phosphorus, which isn't on the graph, and turbidity. This is in contrast to another Vibrio species, Vibrio campbellii, which was more abundant in the dry season and best explained by conductivity. And you can see the relationship between Vibrio parahemolyticus and temperature on the scatter plot on the right-hand side. And this was interesting to us because we are a warm climate. It's warm all the time, and yet Vibrio parahemolyticus did respond to warmer temperatures as well. So this is important information to understand how the different species respond, and with changing conditions, changing seasons, or even longer term global changes, global climate changes, it's important to understand how they will change and make predictions and plans accordingly. So the aquatic monitoring study that we did earlier for Param water was looking at presence absence of Vibrio parahemolyticus and Volnificus, but we were interested to understand whether levels in biota were also different in the seasons, as we had found in the water samples as well. So we were lucky to have a student, David Simmer, a master's student, who just finished his sampling in January and is writing up at the moment. So these are a couple of points from his work, and hopefully he can explain his work in more detail at a seminar later on. So he was interested in Vibrio parahemolyticus levels in long bungs, but also in sediment and seawater. So we had an idea of what was happening in seawater, but we hadn't examined sediment before. And we thought that would be important for these snails that actually live and feed on the sediment. So he's got a lot of data, but some of the important take-home messages are shown here. So overall, Vibrio parahemolyticus was more prevalent in the wet season, which reflected what we had found earlier. And in fact, the concentrations were highest in sediment compared to biota and water. And they were higher by several orders of magnitude. So sediment seems to be an important reservoir for Vibrios. He also screened the samples with the virulence gene VOP, which I mentioned before. He could have tested all of them, but that would have been a nightmare. So we just decided to stick with one. And he detected the virulence gene in one sediment sample. And that was this wet season gone by. In January, I think he did the sampling. And that was after a period of monsoon activity. So the take-home messages there are that sediments are a source of Vibrios that we need to look into further. And it has implications for resuspension events during severe storms and monsoon activity. And again, I can't keep my head around all the predictions for different parts of the world, but I'm pretty sure it was said somewhere that the frequency of cyclones and storms and the intensity of them may increase. So anyway, with all this information, we can hopefully make some predictions about where Vibrios are going to go down the track. So moving on to another organism, tropical rock oysters. There's an increasing interest in tropical rock oyster aquaculture in Northern Australia, and the red dots show the locations where this is currently happening or about to happen. And it ranges from a family-run farm in Bowen to Indigenous enterprises in the NT, and then over to more commercial companies over in the WA coast. And we already know that Vibrios are a problem, can be a problem with shellfish safety around the world. And we felt that we needed to know more about Vibrio ecology in oysters to support this industry that was developing in Northern Australia. So to do this, we worked on a Fisheries Research and Development Corporation project, which started last year. It has a number of objectives, and I only wanted to talk about one of them today, which relates to the previous slides. And that was to look at the Vibrio baseline, so the Vibrio community in oysters, including species that were toxigenic. So we looked in water, and we needed to know what was happening with oysters. So we piggybacked on another FRDC project that had started, and this was to collect wild oysters, black lip oysters, and if they weren't there, also milky oysters, to measure cadmium levels, because cadmium concentrations can also be a problem with oysters across Northern Australia. So we were lucky enough to work with the Darwin Aquaculture Centre people that were doing the sampling, and we obtained some of the tissue homogenate, and we could extract the DNA and send that away for sequencing to look for the whole Vibrio community in these samples. So in all, there are about 13 locations, and these are wild oysters. And we also included farmed blacklip rock oysters from Goulburn Island. And right before the project was starting, we also included farmed blacklet rock oysters from Bowen in Queensland and milky oysters from the Pilbara area as well. This was a one-off sampling event that happened over a 2-year period, depending on tides, and then COVID was in there. So we can't really make any statements about levels of vibrios over time. It was just one point in time to get pretty much a list of vibrio species in these oysters. And in all, we identified 48 vibrio species. And that's right. The most abundant vibrio species are underlined there. So Vibrio parahemolyticus appeared again. That's the yellow part of those taxa plots. It was pretty much present in most of the oyster samples except for the Bowen ones. The other prevalent ones were Vibrio harveyi and campbellii, the orange and the red as well. We also identified Volnificus, Fluvialis and Antiquarius sequences in the database, which are also known to be human pathogens. And as I mentioned, Parahemolyticus was present in most samples and often it was the most abundant vibrio species. The Bowen oysters, which are the ones way on the right-hand side, had quite a different vibrio community. They took a few days to get here. The post office was closed, the plane didn't take off, it was all that COVID stuff happening, wrecking our lives. And so we're not sure whether that different vibrio community on the right-hand side was due to the transport time taken or whether that was real. But like I said, it was a one-off and it was to get an idea of who was in there. It might seem alarming that all these vibrio species are in oysters and they're things that we eat, but they are part of the natural ecosystem. And we have a lot of bacteria in our guts that help us live and stay well and stay healthy. And the same is true of oysters as well. So just because Vibrio parahemolyticus sequences were detected in there, it doesn't mean that you can't eat them. And on top of that, we don't know whether there were virulent strains there or not. So this is something that we're hoping to tease apart, and maybe Karen can explain that a bit more in her section as well. So to summarise, seawater and oysters in Northern Australia have a wide diversity of vibrio species, including the big three. Different factors affect their abundance. For example, temperature, salinity, as well as nutrients. Vibrio parahemolyticus is a major component of vibrio diversity in seawater and oysters, but the virulence genes are not often detected in these environmental samples. And finally, from David's work, sediment may be an important reservoir for vibrio species and is something that we need to look further into. Some of the gaps identified, including the need to develop virulence-specific detection methods and not rely solely on measuring the total species, but just the subset that makes people sick. We'd like to know how vibrio species, especially the big three, in oysters respond to changing conditions. And we're nearing towards the end of a project with the Yagbani Corporation over on Goulburn Island. We've been getting oyster samples from them every month or every couple of months since September last year. And we're interested to see how the relative abundance changes over time and in seasons and in response to any events that happen in that time, such as monsoons, whether there's any heat waves or algal blooms, and whether this has any implications for harvest times. And finally, we would be keen to also set up some trials in the laboratory, just to look at the expression of virulence gene, what makes them switch on, switch off, how do they respond in relation to temperature, changing salinities. So I think that's it from me, and I'll hand over to Karen for her bit.
So mine's a little shorter, but you've done really well. It's only 4. So some of the things that Anna's talked about, I'm going to pick up in these 7 or 8 slides. And what I want to do is talk to you about where we want to pitch this work and on what stage we want to play. So we have an emerging tropical rock oyster industry, and there are very many established industries down the south of Australia. So to continue to operate here, we're going to need to work nationally and with New Zealand to look at this issue. Also, we need to be part of the narrative, because Anna showed you Vibrio parahemolyticus, VP, I've used that acronym throughout, is plentiful in our oysters. And so the issue of virulence is going to come up through these 7 or 8 slides, and we need to be there having the conversation about the regulators can't be just detecting levels of VP as a way to accept or reject vibrios oysters at harvest, because for the north, in our warm waters, that would kill the oyster industry before it even gets started. So this is what our, these 7 or 8 slides is geared to do, and I'll be really keen on your feedback too. So if you think about the environment in which we're pitching this national next stage, Anna is going to want to do lead research more locally, but we also want to do something nationally. And so we have to explain this for oyster farmers, regulators, so food safety folks, people in fisheries, aquaculture, and it's not an easy sell. It's quite complicated. And so we're trying to navigate our way through using an approach that makes sense to different people in different sectors. So we're going back to basics, and Anna's touched on this, is understanding what it means to talk about an oyster microbiome. And I'm about to do next month a children's university session where we talk about your perceptions of bacteria. And I know most people think about bacteria of the really yucky things on the top of your oyster, on your yogurt container in the back of the fridge. It's always the really gross stuff, and we have to get across a message that it's not just about that. So for example, with the oyster microbiome, there's so many ways that you can get bacteria in the oyster. They're passed through from from the parent, the larval environment, and the larval microbiome. It's naturally occurring in seawater, as Anna said, so they'll get it in via filter feeding, and it might just be a transient association. It's just passing through. So they're harvested, they close up, and then whatever's in them might have just been transient, or it's more than that. It's retained in the oyster, and it could actually cause disease or a gastro for us or for the oyster, or it could be an opportunistic pathogen, as vibrios are, that circumstances out in the big wide world are not pleasing, and so they get into a host to be safe. So, it might be anthropomorphic, but it's kind of a fun way to talk about bacteria, in that they don't always like where they are, and so if they can get in, they will. And they might have no measurable impact on the oysters, but they could have an adverse effect on humans. So there's so many ways and reasons. But if we understand how VP, parahemolyticus, which is the one that closed down Coffin Bay and the Tasmanian oyster industry for a period of time, if we understand how it invades, persists and causes illness in humans, it'll give us a chance to make inroads into shellfish food safety. I'm having trouble with my words. So you ask green sharing's in the way of my titles, but what it says there is Vibrio naturally occurring and very adaptable. And that's not news now, but it raises a lot of problems. They're incredibly successful. They grow really fast, one of the fastest growers. They're really adaptable, and they have not 1 but 2 chromosomes just for that extra zing on gene opportunities. They use environmental signals really well, and if they want to and circumstances are right, they can just bloom, and very responsive to the environment. As Anna said, some but not all are virulent, and if they are virulent in the right circumstances, they get access to a rich host environment and outcompete their buddies that don't have those virulence genes. As I've said, they can accumulate in animals with or without disease, and they're on the move. They can swarm, so think of bees, but literally with climate change, they're moving, and that, as Anna said, that brings both stress for them and opportunity. And so the problems, and this is something that I think we've got a lot across effectively when we were in Adelaide meeting with oyster growers, regulators. It's really clear that a naturally occurring organism is much harder to manage than something like a sewerage incursion into an oyster harvest area. That's really obvious. You suddenly get an increase in E. coli or fecal coliforms, and you can shut it down until it's clear. But this is not associated with a pollution event. We don't know enough about the environmental drivers of abundance and virulence, and I'll come back to that several times. And as you know, climate change is bringing and has brought significant uncertainty. The current tests for Vibrio parahemolyticus numbers are inadequate measures of risk because they don't address virulence. And just sitting with numbers of bacteria is really dangerous for our northern oyster industry because they occur naturally in a watery environment that's high in parahemolyticus just because it's warm. Well, that's one of the reasons. So what we need is fast, accurate, virulence-specific and accessible detection methods. And what became clear to me with the meeting that was with oyster growers is at Coffin Bay, where there were 48 growers in one bay, there were so many problems with the local labs. They didn't know what method they were testing. They, just as growers sent it to them, they didn't get results that made sense. People were still getting sick and it was being detected. They couldn't detect it in oysters or water. They didn't know really what the commercial labs were looking for or how they were doing it, sorry. And so the whole bay got shut down. So all of the growers, even though it might have been just one or two or post-harvest. So this is really critical, but as I explained to you, it's very loaded. So environmental drivers, climate change and virulence, I'm going to run through this fairly fast. Surface sea temperature is the most common driver of abundance, but salinity can also be important, and this is worldwide. Different strains within a species can respond differently to changes in the environment, so that makes it even more complicated. We do know oysters host higher levels than other seafood, and there's that phenomenon of hot oysters where they're perfectly healthy, but they just host a really high level of BP, and so you can miss it. Climate change, we know that it is going to affect a whole range of attributes around Vibrio parahemolyticus and others, but we really are behind the 8 ball worldwide. We really don't know what's going to happen. We just know it is changing. We don't know how to predict it. We do know that marine heat waves exacerbates disease outbreaks in oysters, and one hypothesis that's been put forward is that in environments where the vibrios are moving to that they haven't been before because of warming sea temperatures and warm currents, they get in this new environment and they suddenly, forgive me, I think of bacteria as little people. They think that they're, oh my God, what am I doing here? I have to get out, I'm panicking. The virulent strains can get into a host. It's a rich, safe environment. And so if they can, they will. So in these climate change or stress environments, they're actually causing disease outbreaks just to get away from the environment that wasn't suiting them. Virulence, as Anna said, it's linked to the TDH and TRH genes. And also, as she mentioned, there's this T3SS secretion system, the injection thing. But clinical samples can lack all 3 of those. So where does that leave us if they're not good indicators? And so in the literature, there are things popping up all the time that might be virulence genes. They might be useful or maybe not. And it's just a very confusing story. And as Anna's mentioned in our experiments, that just having high levels of VP doesn't mean that there's a pathogenic subpopulation present. And that's quite a hard sell. That's quite a hard thing to explain, because everybody tends to associate high levels of a bacteria with problems, but that's not the case necessarily. And that's a message that we're trying to get across in that FRDC project that we're partway through. And there's also the issue of pathogenicity, whether we need to be looking at RNA gene expression as well as DNA genes. Just having a gene doesn't mean that it's being expressed. So we do need to think about that. But with all the other unknowns, that's sort of one that's getting lost in the mix. And as I've mentioned, there are still very few studies worldwide on ecological drivers and how they impact on these virulence genes. So just taking stock, the drivers of abundance are not the same at all locations. Virulence is complex. Climate change brings significant uncertainty. Now, we've said that we'd like to understand environmental drivers in our part of the world, and other groups do, wherever oysters are grown, for example. However, this is a map of Australia's complexities around currents, eddies, and upwellings. And if you notice around South Australia, there's some little blue markers that are seasonal upwellings. And there are persistent eddies, there are currents throughout. This is a really dynamic system. And if we know that vibrios can move on currents and might be coming up in upwellings and eddies, how do you make sense of that if you're going to try to have an argument that you need to go to each location and understand the drivers for that particular jurisdiction? It would be, excuse me, this is a great opportunity for vibrios, but what I've said down here is basing national food safety guidelines on the environmental drivers for one location is clearly going to be problematic, given how dynamic our environment is. And I'm going to propose in the next couple of slides that we need to think big and embrace technologies, not because they're cool, but they are, but because they give us our best chance of staying one step ahead. And for some of you, these technologies are well known, but think about sitting in a room with oyster growers. So putting forward a proposal, which we're co-leading with folks from University of Technology, Sydney, New Zealand organisations and University of Tasmania. And we're suggesting a process that has multiple steps, and that is looking at how VP becomes a human pathogen. And it's all based on what virulence genes are responsible so that we can have better screening tools. And then we can go back out into all those different environments and look at upwellings and eddies and currents. So I'm going to not talk a lot about this because it's in the next slide, but this is a whole genome sequence phylogenetic map, but I'll talk about it a little bit more. I'm promoting that we have a national and New Zealand approach to this, and suggesting that we work with health departments, if they'll work with us, to get access to clinical samples or clinical isolates of Vibrio parahemolyticus. Similarly, oysters and other aquatic foods, and get those isolates, sequence the whole genomes, and then do a phylogenetic analysis to look for virulence genes. But where we differ is not only that we want to use not just clinical but animal, but we also want to use environmental samples. And I've got a whole lot of symbols there around monsoons, cyclones, heat waves, around oyster farms, sediment churn, salinity changes, looking at water and sediment samples during all those very key times that we know are associated with changes in the vibrio. So also getting samples of those environmental isolates. Looking at the complexity of those using the whole genome sequencing, as I said, the next slide will explain that more. But then looking at potential virulence genes based on that whole genome approach and developing assays for particular virulence targets, both in water and with oysters, to see how they perform and if they're potentially good targets for routine screening for a whole range of groups nationally and with the New Zealand partners. So I'm going to go back over this, but let me look at step one, identifying genes common to clinical samples. This is actually from Miriam Kessley, sitting in the back there, who's worked on, she works 1 day a week at the Menzies School of Health Research and the other time with us. And so it's a really lovely collaboration. And this is a whole genome sequence map for the melioidosis bacteria, Burkholderia pseudomale. And I showed this to the room full of oyster growers and food regulators. And I said, this is really similar to our situation with Vibrio. They are opportunistic human pathogens. They occur naturally in the environment. And the way Miriam's done this phylogenetic tree based on whole genome sequences is that the outer rim with the grey are different times of years. The inner circle with the different colours is different regions. And then within that, you can see the stars of different colours. So that includes isolates from animals, clinical, the red, environment, soil and water. So you can see it's a perfect example of what we want to do with Vibrio parahemolyticus, and we're really well placed to do it. The other good thing is that traditional sequence typing, particularly when there's an outbreak and you want to track the epidemiology of an outbreak, when you do have a repository of whole genome sequences, rather than some really labour-intensive laboratory trials, it's a one-line sequence code. It's an R code, and you can do the same thing. You can do the same thing with SNPs, which is good because we've got Deb Holt in the background who's doing some fabulous work with her student on SNPs. Again, R code, once you've got that information. You can look at the sequence typing of the whole genome, the core genome. It gives you a huge capacity to compare and contrast all these different sources, environmental, animal, and clinical. You can look at spatial or temporal clusters, as you can see there. You can compare clinical to environmental to investigate virulence. You can look at patterns between virulence genes and pathogenicity, and you can investigate virulence potential over space, time, and host. And it's easier to screen whole genome sequences for non-virulence genes, again, because it's R code, than by qPCR. I'm not discounting PCR, you have heard of that around COVID testing, but in the early instances of this process, this is a much better way to go. And it gives you power to visualize and analyze a whole range of functional genes. So once you have a database, so many other groups can work with it, depending on their interests. So it's a very good collaborative tool as well. And you can go back in time if you want to. So it sets up a legacy beyond the life of the project. And it was pretty well received, a little bit of stunned silence. It's not new, but with that group of different sectors, that audience, it was a little bit shocking. Step 2 is to test gene responses to environmental drivers. So this step 2 in what we're proposing is some environmental challenge tank trials using selected B-pre strains. So they're selected based on their clinical profile using whole genome sequencing, which is step 1, and focus on key genes that are associated with virulence, but not necessarily limited to virulence. For example, the ones we've already talked about, but there's a whole range that could be used depending on patterns that emerge. And try to understand how those key virulence genes behave in these tank trials. We need to be really clear that we're working with something that's relevant. So we can look at presence and abundance of our target genes by qPCR, or look at gene expression, which means you start with RNA. And so we use both plus or minus shellfish. We can actually look at different oyster genetic lines as well. So that's where it becomes truly national, and the tropical rock oysters as an emerging industry doesn't get left out. And we can also manipulate temperature and salinity and see what happens with those particular genes and whether they'll be useful. And we can measure responses, presence, abundance, presence versus abundance, and gene expression, and also the whole bacteriome response. You can look at the whole bacteria as well and see how they're responding if you want to broaden the work. So understanding the influence environment on VP virulence genes will give us access to screening assays that give us the best chance for early warning food safety risk. Now, this is in an environment where commercial labs are very conservative. The regulators are very conservative, and they're still using colony forming units, which is how many colonies you can grow on a plate or in a tube. And in some cases, they're just rejecting the notion of actually looking at abundance at all, just presence, absence. And so we've got to move out of that system and into something more radical. And that's not going to be an overnight job. The third step, and I'm nearly finished, is that we need this accurate virulence-specific detection in oysters. We need, currently quantification is by this most probable number or colony forming units, but that doesn't work well. It's a culture-based approach that doesn't work well where the vibrios are no longer culturable. They form those shriveled spheres that Anna talked about. So if they're still potentially pathogenic, but they're not going to be cultured, then maybe that's why you get nothing when you do a culture approach. Just quantifying them doesn't tell whether or not there's pathogenic subpopulations. We have the hot oysters that I mentioned to you, so it's very erratic. And quantification by direct detection without the culture step would be a really great approach if we found these virulence targets. But unfortunately with oysters, direct detection levels are really poor, because if you can imagine how snotty an oyster is, the DNA coming out of that, a lot of times that actually quenches or inhibits the PCR tests. So virulence gene detection pros and cons. We'd like to promote, if we find that really good virulence targets and it performs well in tub trials, these little mobile handheld PCR machines that would be really fantastic for environmental surveillance. And if we've got the right target and we look at direct approaches, it means that if you don't have to culture, you can freeze the samples, which is really great for remote communities, or if you want to batch a whole range of samples in the freezer. Right now, you've got to get it to the lab so it can be cultured. We can do direct presence absence detection of virulence genes using these mobile approaches pretty easily. The problem is if we want to get to quantitative detection of these virulence genes, but I don't feel that that's necessary straight away. So if we could get to that endpoint in this national project, we would really be in a great position to support the industry and try to encourage commercial labs to consider using these environmental approaches. So putting it all together, identify genes common to clinical strains, test their responses to environmental drivers, develop assays, and then work with the regulators or those folks involved with quality assurance to juggle time and cost, the producers and how they're coping with these added burdens, and food safety issues. Right, so all these people, we've got the old version we left off. We've got these fabulous people that have helped us. The Fisheries Research Development Corporation has been a great support, and we hope will continue to be a great support, but also the Darwin Aquaculture Centre, Matt Osborne and Sam Noland. And also I had there, but Cynthia Coyne prepared the flyer that Anna showed for our FRDC project. She's our communications leader for that project. So thank you. Sorry.
All right, thanks, folks. That was really interesting. So well, if you're playing at home or in your office, if you type us a question, if you have any, otherwise people in the audience, I'm going to march around with the microphone. Do we have any questions following that? Hey, there we go. That stops me kicking off. Yeah.
Thanks, Karen and Anna. Obviously, the vibrios are not carrying these virulence genes to cause infections in humans, because that would be a waste of time. And you did say they do cause infections in some other marine organisms. But is there any evidence of causing disease in any of these shellfish?
Yeah. So apparently in Thailand, the prawn farmers are having a terrible time with Vibrio parahemolyticus. I'm not aware of Vulnificus doing so.
Yeah.
In the hatchery, Zara, who's online, her PhD, mortality events for larvae tend to be a different species. So no, not commonly.
Okay, any other questions? I've got a really specific one. Hey, Anna, in one of your slides, you were talking about the samples from the 3 creeks around Darwin, and you're talking about collecting nitrate and phosphate samples. Do you know, well, I don't know, this might be too specific. Were the samples in the creeks, or was it out offshore? And I ask because we're talking about this in another project about challenges in assessing nutrients in seawater, where salinity, I think, is an issue for the labs.
Well, we weighed it out into the water and collected the water sample like with a pole. So we went into the water a little bit and then collected it with a pole and sent it away for total nitrogen and total phosphorus. And we sent it to labs in Queensland that do that sort of analysis.
Okay, we'll have to look into that.
We've had problems with nutrients with effluent samples. That confuses labs because the levels are...
Yeah, recall, there we go, not yet. I just recall people talking about, I think they had to keep diluting the samples till they were below a salinity threshold before they could analyze anything, and then you can't detect what you're looking for anymore. Hey. Ask them a real hard one.
This was a great presentation of presentations. Thank you very much. Just a quick question. This being an opportunistic pathogen, also being called an opportunistic pathogen, have there been any studies on relationship of, say, clinical presentations and also host risk factors? Like, have these all been healthy people? And could it, for instance, be that some clinical strains have virulence or the virulence genes and others have not? Could this also have been a difference in the host risk factors, that some hosts just were immunocompromised, for instance?
I think anyone can get gastro from those Vibrio species, whether you're immunocompromised or not, so you can get it. And maybe for people that are less healthy, it can develop into something further. But I haven't read anything about that sort of relationship. No.
I had the impression from Bart's work that the severe outcome from one of those 3 samples from MacArthur River, that person did have some comorbidity. That was Vulnificus.
I think for wound infections, definitely, but for gastro and eating not really related to people with underlying conditions.
All right, so you can see, thanks, Cara, for the essay. But it's mentioning, is this a, oops, hopefully that's not a comment back to me, sorry. So the citizen science, is this something that you can look into to facilitate the project? I don't know what EPI data is, but is that something that you can use to join the dots for climate change impacts? So is citizen science something you can do to help your data collection, I guess?
Well, the other question was how soon after a marine heat wave are there incidents in hospitals? But I don't know the answer to that one, but what was really interesting was that, can I tell that story? That after the big January monsoon, when David Simmers, the levels he was detecting in the long bums just shot right up, Anna actually called territory pathology and mentioned that we were getting really high levels in the water, or in sediment, sorry, and the long bums. And a couple of days later, clinical samples came in, so they contacted Anna. So that's actually formed a bit of a bond between the two groups because of that prediction. I don't know the other. I think in answer to Cara's other question, it's early days. If we could find some targets and give the oyster growers confidence that we can detect it reliably, plus or minus virulence, because they got that, then I think they'd be happy to do some citizen science with us. They're a bit suspicious at the moment.
There you go. All right, so we got one last one. We'll do last question and then we'll wrap up.
Sorry.
You're right.
Thank you. That was great. I was just thinking back to very early on, learning about suspension feeders and the fact that things like mussels and oysters are very, very good at concentrating metals. And that used to be the main concern, I think, about eating filter feeders, suspension feeders, was metal contamination. So are you seeing any interaction at all between metal concentrations and vibrios? Has that been looked at?
No. They kind of, I think the reason is that we don't know, because in that, you know, that slide of all the samples from the north, we don't, because most of them are wild, we don't know how long they were out there, and they kind of max out with the cadmium for them, I mean. And so I don't think it keeps going up, and I don't think there's a good relationship. And to do that, I think it would be fascinating to have them come from the hatchery, the nursery, and out, and then track them for both. But we haven't had the opportunity to do that. I think the focus for us is for fisheries. For the first commercial sale of tropical rock oysters, will be having in place the Australian Shellfish Quality Assurance guidelines for the region, and ours are going to have to be different. And so their focus is on getting all those blocks in alignment. And I think some of these more subtle questions, and even vibrios are not yet in the Australian Shellfish Quality Assurance standards, but they will be soon. So I'd love to do a study like that. I think it would be really interesting. Especially that virulence metalloprotease, that if virulence is associated with metal binding, zinc and cadmium work as antagonists.
Excellent. All right. Well, so thanks, folks. That was a great presentation. Can we give them one more round of applause?